Okay, I guess that's going. Okay, all right, here we are. Because we've got two more, okay? And believe it or not, you're like, forever. This one we'll spend a little time on, but this one we don't spend any time on. Actually, you want to hop down and do that one, or do you want to save it for last? Save it for last. Okay. Okay, so this one gets around with false feet. This one gets around with a single flagellum. Even if they're in a colony, they still only have one flagellum. So now this one is Ciliophora. And here is that phora, again, right, means bearing. And this is cilia. Cilia, bearing cilia. Okay? So how is this different? So, all right. This was our euglena. So let's take this off. There's his eye spot. This is a food vacuole. No, that's a chloroplast. Anyways. All right, here we are. Ciliophora. Now, if this is our euglena, okay, if that is our euglena, this is our paramecium. Okay? Much, much different in size. I'm going to say this just because I have forgotten to say it in past classes because I think it's kind of obvious. I'm going to have you do an exercise in your journal this week to give you some practice doing this. But I did have a student, when you start to do this lab next week, you're going to have a page that's got all these circles on it, okay? And you see something, you're going to draw me a picture of it. So you're going to draw me a paramecium. Okay, there he is. And you're going to try to label, any parts that you might see in there. And let's say that this is on 100 power, okay? When you turn it up to 400 power, what should happen? Say it out loud. It should be bigger, right? If this is 100 power, then 400 power should be like that, right? About 4 times as big, right? I actually had him, like, this was their paramecium at 100. This was their paramecium at 400. Why? Why did they do that? I don't think they had it confused. Maybe they did. Maybe they reversed their calculus. Just make sure that if it's this big here, then it's going to be, this big here. Right? I'll have you do an exercise this week. So, here is our paramecium. Okay? He doesn't get around with the flagellum. He's got hairs all the way around him. Okay? All of these hairs, and it's unlike those, what are the ones called on the bacteria? Fimbriae, right? Fimbriae. Can't remember. Fimbriae. What was the interesting thing about the fimbriae? Did they use those to move around? They did not, right? They had the flagellum to move around. In the paramecium, this is called cilia. Okay? You'll see this word again, okay, because there are cilia in the human body when you start to study that, right? In the intestines, don't we have cilia? There's somewhere inside that we have cilia. Drawing a blank. But these hairs are actually used to move, okay? And they move very fast. If you think these guys move fast, you would think being that big, they can't get around. But they do. They get around very fast. So a few things that they have that the other ones don't. Okay? They have a nucleus, Okay. This one is the big nucleus, so it's called the macronucleus. Macronucleus. The reason you have to have a big nucleus here is this is helping to run all of these hairs. Again, this makes it look like he just has a line of hairs. They're three-dimensional, okay? And there's hairs all the way around them. So it's not just on one edge. It's like, and all of those things are running. And for every motor that runs, you have to have a control panel, okay? So it's got a big nucleus running all these hairs. It also has a small nucleus, usually right close to it, called micronucleus. Right? If you have a macro, then you probably have a micro. That's what's used for reproduction. Okay. Let's see. What else do I want to tell you about? He has his contractile vacuoles really look cool. They kind of look like stars. Okay, kind of look like that. Not that bad. Okay. If you see something that looks like a star, that is a contractile vacuole. What does the contractile vacuole do? Regulates water. This is to make sure he doesn't blow up, right? Explode. Then you're going to have his food vacuoles. Those are the same. The biggest thing that this guy has, and it's really hard to draw it on paper. Once you see it, you're going to be like, oh, that is so cool. He has this like indention. Think of it as, if you have a basketball that's not completely inflated, you can kind of dent it in, and it holds that dent. Okay, think of that dent as running down like this. Okay, and like there's this dent area. Hard to. Anyways, there's little hairs that line this whole area. Okay, and they're just washing water down this channel. Okay, down this channel. And then out here at the end of the channel, it kind of has it differently. There's like this little pocket, okay? And here's what it reminds me of, okay? It's washing this water down there, and as food gets down to the end, it begins to blow up like this little balloon kind of like thing, filling it with food. And as soon as it's full, it pops that off, and that becomes a food vacuole. Right? There's our food vacuole. So it goes down the pipe, fills up this. How many of you have ever done those? They come in little tubes, and it's kind of like plastic, and you stick it on the end of a straw, and you blow through the straw, and it makes this plasticky, that's what it's like. There's this little film at the end of this tube. He washes this food down in there, and it expands, and then it pinches off, and it becomes the food vacuole. And you can think of that. It should help. So it goes down. This is called the oral groove. Oral meaning mouth, and groove. Okay? And as it gets to the end, okay, it's going to call this area down here the gullet. And this is called the core. And then that's what makes the food vacuole. Okay, this oral groove is also, these are the first ones that we've experienced that reproduce sexually. So two paramecium are going to line up with their oral grooves lined up and exchange DNA with each other. Okay, and then they're going to separate and immediately divide themselves, producing two new parameciums that are made with a mix of the two DNAs. Okay. So it's slightly different, right? They do exchange DNA, so therefore they're slightly different. The new one will be slightly different than the old one. All right. Let's see. Which ones do we want to remember? What does he tell us? Valentinium coli. Valentinium. Here, this is your pathogen for this one. Pathogen. Valentinium coli. Okay, lives in the gut of pigs. Okay, then they can be passed from the gut of the pig into the, this is going to sound terrible, okay, into the pig poop. It can potentially get into water, and if people aren't cleaning their water enough, you know, you can pick it up. Remember, these are microscopic. It's not like you can look at the water and go, oh, it's got pig poop in it. It's not like that. You wouldn't see it. It would look totally clear. Okay. But how could you get rid of it? Let's say for some reason you are in this country and you're thirsty and oh my goodness, I have to drink some water. Is there anything you can do? Boil it. That's right. I would boil the water for say 15 minutes, right? Probably not have to be. Probably 5 minutes is long enough. I'd probably do 15. I'd probably boil it for an hour. Oh please, I don't like anything. So yes, you can still, or many times they have little chlorine tablets, bleach tablets, and you can drop that in there and the bleach will kill it too. So.
Mrs. White?
Yes.
This might be a little late, but I just sent you on the chat. I just sent you a video. I found a video of paramecium.
Okay.
And if you wanted to show that to the class or just not.
Okay, let me see. Let me see if I can. I have to get through this one more thing. Oh man, my battery's gonna die. It's gonna be really sad because then that's my internet. If it suddenly goes off, it's because my phone is dead. What time is it? It's time to go, right? It's okay, I can't show the video right this second. But let me get to this last one, okay? This one is Protozoa. Protozoa, okay? You don't move. You don't. That's what puts them in this category. The pathogen here is the Plasmodium. Yeah, Plasmodium. And it causes malaria. Okay, Plasmodium is your pathogen. All right, I think that's it. Next week is lab. Journal. Your journal assignment. Oh, that's cute. Your journal assignment is to find something that you're going to draw in your journal, and I want you to do one like as it is, and then take like a hand lens. You have a, what do you call it? Yeah, a hand lens, something that you can look at it closer and draw a close-up of some area of it, if that makes sense. So maybe a picture of the leaf. It didn't have to be a leaf. Draw a picture, and then with a hand lens, look at something closer and draw me a circle of the close-up, just to get a difference of here it is, close-up. Okay, thanks. We'll see ya.
Bye.
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 Bye, Clayton.
Bye.